pretty much set up. Co-pilot's going to finish getting the lighting
they're reading
adjusted.
News. Breaking news.
I think
We're
we're
on
ready
the
to proceed
C4.
with the dive.
Roger that. Thanks a lot. And we're ready to proceed with the dive.
This is dive number 10.
Hey, look at that. Is that a black belly rosefish?
Might be.
We'll take a look.
We have not seen one, or at least I haven't been at the monitor when we've seen one.
452. 438. 453.
See if I can touch down
coral
without
rubble.
startling
I was
him.
just going to say that. Yep, what we expected to see here.
And we haven't seen one of these yet, the whole expedition.
I was
if
wondering.
it is the
Wanna go
black
in
belly
partial?
rosefish. They're very common in Lophelia areas, so we'll probably see more of these.
Okay, video. I have all my swing arms out. Let me know if you need me to change
Looks
any
like
lighting.
he hasn't had his Sunday morning coffee. I know how you feel.
Okay, go in tighter. All right, everything good, Fernando?
Everything good? Everything
Yeah,
ready?
even though
Coming
this
down
fish
a little
is
bit.
red, I believe the reason it's called
Setting
black
up
belly
on
rosefish is if you were to
Maybe
fillet it,
12 delta.
its belly is actually black color.
Okay. We can go ahead and come out, and why don't we go ahead and get a
Was it surprising
30
not
meter
to
move
see
uphill.
him in the
Bearing
Stetson
250
Banks
250
area?
is fine for now, yeah. Right,
Same
Fernando?
kind
Look good
of
to you?
Yeah,
kind
can you
of
bring me my
substrate
sonar a
and,
little bit,
under
navigator,
the
please?
Gulf
Understood.
Stream? Is that
Decreasing
just-
range.
Yeah,
on
I'm
farther
Sirius
surprised
south
we didn't
to
see
50
this
meters.
guy. We
Copy,
were
thank you.
deeper though. I don't know what the depth limit is for.
Maybe
This
so,
fish.
yeah.
Bridge is RV nav.
Because we saw them at the Charleston Dome.
Yeah, we're safely
Yeah.
on bottom and ready
Quite
for
a
a move
bit.
when you are.
Okay, great, I'm ready.
All right,
It was-
range 30
deeper than
meters,
this dive. I
bearing
can't remember
250
exactly the
degrees,
water depth.
speed decimal 2.
That's 30 meters, bearing 250, speed decimal 2.
So
Good
we
copy.
are at a depth of
fish.
450 meters. And we today marks our 10th dive of this expedition. And we're located on another of these coral rubble mounds. It's known as Lophelia Mound, or Cape Fear Lophelia Mound. And it is part
Go partial
of
video.
the
Move initiated. Thanks,
Cape
Bridge.
Fear Lophelia Mound
Hold
Deepwater
that.
MPA, Marine Protected Area. See a pencil urchin in the lower left and the Lemonema fish.
Okay, let's come out.
Lemonema melanurum, I believe.
I'm going to go. Ooh.
And our dive track today
Bunch of
is we will
squid.
be heading
Yeah,
into
I'm
the
going to
Gulf
tilt up.
Stream's current,
I think they're gone already.
which fortunately isn't as
It'll shoot
bad
off the
as
starboard-
it could be.
starboard side, so you can't-
And we
It's
will
out of-
be
Yeah, we're seeing a lot of squid here
slowly
today, Washley.
elevating ourselves into shallow waters with the peak being the top of the mound. But we're going up a flank of the mound. It's just a long, extended ridge that moves off the edge of the mound, which is probably formed by the Gulf Stream's current flow around the main body of the feature. And Lophelia,
Oh, yeah?
the stony coral, probably took advantage of that location. Is that a squid on the bottom?
Yeah, it looks like it.
Still floating upward.
Can we get a zoom, pilot?
Yeah, sure. I'm going to come back down here.
Thank you.
And what are you looking at? What, you
Bottom
want to
there.
see the squid? Gotcha.
There he goes.
Oh, he's gone. There was a
Yeah,
Is
we've
it a-
seen a lot of these? If
What
you
is it a
tuned in
school
as
of
we were
squid? What is
descending,
it?
We had a whole
I
school of
think
them.
it's
I don't know if we have a
co-
school of
copepod.
squid.
Squid
school of squid? Is
schools
that correct?
grouping.
Our colleague Stephanie Bush, who's a cephalopod aficionado, said it is a school of
Okay, good.
squid. A squid school.
Well, we had a school of squid swim by just a couple minutes ago.
That's another squid on
Saw a
the
lot of
upper
them in the
right.
water column as well.
Northerly Menimas.
Yeah, the squid were definitely keeping us company as we
How long was
got
that move now?
further down
It's
and
a
closer
30-meter
to
move.
our start
Copy,
point.
30
I
meters.
think we were scaring the
We
ink
are.
out of
Your
some
ship
of
has
them.
moved 15 meters, so halfway through the move, looks like.
There's a lot of ink
Copy.
in the water as well.
All right.
70 degrees on the rotator.
Copy. I'll push ahead.
So Cheryl, these white stalks are Lophelia, I assume.
Yep, that's what I would imagine. Oh, look, a little cat
what's
shark.
that?
Yes.
Nice looking. Is that a
Nice.
shark? No.
Yeah.
Oh, good. The fish are keeping us company because there isn't that much else in the way of coral and sponges
Should
in
have
this
touched down
environment
lightly
yet.
and not
We
disturb
do
them.
expect to see a lot more.
So I think this is actually the chain dogfish. Ooh, and that's a different looking fish too.
Look at it, 3 in a row.
3 in a row. We can just look at everyone. Oops. That little guy's not cooperating as well. So I think, is this a chain dogfish?
That's what
Okay,
I
let's go in video.
seen.
Yeah.
Called, yeah. And again,
Hold
they're
that.
common on the Charleston Bump area. They're beautiful. I love their coloration.
Very cool. Okay, you want to zoom in on the head?
Let's see his
Can you bring that?
gills on the side. Nice flattened
No. It's already
head there.
75 degrees.
And forage along the bottom.
What do you need?
You want me to stop the ship?
Yeah, sure.
I think we're okay.
Okay. All right.
Good morning. Do we have someone on the line?
How much is left in the move?
7 meters.
Yeah. And it's flat, Fernando, and I can move ahead pretty quickly if I need
All
to.
right.
Cool. Okay, let's come out.
I have selected waypoint 2.
Copy that.
There's another view of the black belly rosefish. Different one. We've seen several of them already.
You know, it'd be cool. So you have the rotator. You can rotate all the way back.
It would be neat. Or see at least directly down. At least, right? Point the camera down 90 degrees.
And what we're looking at is a field of coral skeleton rubble, which
So that way
makes
there's
up
never
the
a blind
entire
spot.
framework of this mound.
Are you talking
It
about
is about
B2 or
Serious.
series?
at least
Oh,
40
okay.
meters
Sorry, I
in
mean, I was
total
just speaking
relief.
of that in
Probably
context, yeah,
more.
serious
We're
rotator.
starting along a
So that
flank
way,
of
like,
it, maybe not at the
You
greatest
took a
depth
big, you should
at
have
its
Sirius
base.
looking at you. The big Zeus looking at you.
Another one of those same fish.
And the brown material is that rubble. And you see individual white stalks scattered throughout. Those are more recently dead. And any of this hard coral can act as substrate for organisms if there's enough food for them coming through the currents here.
Bridge move
You
complete.
can come up a little bit, Fernando.
And
When
if
I'm
it's
directly
stable
below
enough,
you,
Move complete,
it should
thanks,
be
Bridge.
like 17 or 18 meters.
Copy that.
It's like that move's complete, pilots.
Copy.
Probably not a lot of layback today. Almost 0, so. Or, swing.
Lighting looks good, Bob? Okay.
I think the drawer might be open a little bit.
Yeah. Yeah.
Oh, yeah,
That's
you
from
never
when they
bumped
cracked the
it in.
boxes, yeah.
Yeah.
And this is a coral
Urchin
hake
there.
that you're seeing now, Lamanema melanurum. Just going off the screen to the right. Another
Tyler,
pencil urchin.
can we take a quick zoom on the pencil urchin,
Sure.
please?
Okay, go ahead.
Wow, see all those little brittle stars too. Every little nook and cranny. It's interesting how some of these pencil urchins have one or a couple red spines. sometimes.
And they look like they're adapted for other things, maybe maneuvering over the terrain.
I'm not sure that that's known. But yeah, some of them don't have those also. So
Okay, we can come out when you're clear.
all those little orange arms of brittle
Is the
starfish.
anemone in view? Oh, there it is on the bottom left. Okay, go ahead, video. Squid and Sirius camera. Keep seeing him swimming by.
See all the brittle stars. They look like a little forest
Copy.
instead
Yeah.
of individual organisms. Is that an anemone?
Yep.
Individual. And they do migrate around, not very quickly.
Some of them are quicker when they hitch rides on hermit crabs.
Yeah, for sure.
Okay, come out.
Wasn't supposed to.
I see it, the red one up there. Okay, go ahead, video.
This pencil urchin doesn't have any of the red spines, but his test looks more red also.
Go in tighter if you got it.
And that's
Hello, Mike
sargassum.
Becky on here.
Hello, Mike. How are you? Good morning.
I'm okay. Good morning. I'm operating on my cell phone today and doing a honeydew project.
Oh, wow.
But I just tuned in and saw
Okay,
several squid swim past as you were getting close up on that chain dogfish.
Yeah.
And then I heard the pilot or somebody, one of the operators, comment that there were squids in the Sirius. I don't know if anybody's told you anything about those squids yet.
No.
They're short fin squids.
Okay.
That's the genus Ilex. They're probably Ilex elisabrosus, which is a really important commercial species and also really important in food webs. But there is, in that area, there's a second possibility that is very difficult to distinguish from elisabrosus. Ilex octagonia is reported from the area you're operating in. So I can't be sure about the species, but what you're seeing is short-fin squids of the genus Ilex, and they achieve very high abundance sometimes. Sometimes you see very large schools of them. I haven't seen any large schools yet, but I have seen some small schools since I've been tuned in.
Actually, right before we got to the bottom, there were, I don't know, at least a dozen cruising around.
Yeah, when I say large, I'm talking hundreds to thousands.
Oh, okay, yeah, not that large. Oh. Thanks for putting that into perspective.
Sometimes you see in those kind of aggregations, but
That's a squid
not
university.
At all unusual to see a few to dozens of them.
Wow, that's great.
Okay, I just thought I didn't know if anybody else had said anything about them. But,
No,
thought
thanks
I'd
very
key
much
in on
for
it a
that.
little bit.
And hopefully we'll see more as the dive goes on.
Okay, I'll try to continue monitoring on my cell phone, and then if something interesting cephalopodish comes up, I'll call back in.
Great,
Take a
thanks,
look at
Mike.
this coral here.
All right, talk to you later.
Thanks, bye-bye.
Happy Sunday.
Bye.
Our first live Lophelia. It's a scleractinian coral that's responsible for building these mounds over thousands of years.
Okay, let's go in video. Thanks, Fernando. You're on top of the lasers.
You should almost have them on like a momentary So you just have to hold them.
Yeah. Yogi's nice because it just has a button on top of the joystick, so the pilot just goes
Cheryl, does
on and
this
off, you know, at
species
will.
have, or any of the stony corals, do they have a certain
Go
number
tighter if
of
you got it.
tentacles? We have the Octa Corals that certainly have 8, but is there a certain characteristic number?
Yeah, they're in the hexactinellids, so they'll have 6 tentacles or a different variation on 6, 12, or something like that.
Okay.
So
I don't think I'll count in this little current. It'd be tough to.
you could almost count.
You could, yes, in the lower left, maybe.
Yeah, it's a beautiful shot.
And are we looking at hydroids that are also living on this coral? Or?
Those little feathery things that are sticking up right in the middle of the screen are hydroids.
Okay, you can come out.
And this is a fairly small colony.
Probably get a move in.
And what kinds of growth rates
Okay, just do 30. Yeah.
do
Understood.
they have in terms of
Still 250 for now.
how long does a single polyp live, and how long would it take for the organism or the colony to grow
Bridge
to
RV, yeah.
that size? perhaps?
I don't know if we know exactly how long.
Yeah,
a
like the move
polyp
range
lives.
30 meters, bearing
Sandra
250
Brooke
degrees,
has done
speed
some work
decimal
in the
2,
Gulf of
same as last
Mexico,
month.
where she brought a colony up onto
Good
the
copy.
ship, stained it red, and then put it back down and went and visited later. And different branches grew at different rates, so it's really hard to get like an absolute
You want to do a partial zoom?
age.
See that thing waving around?
But it was on the order of
Hold that.
several
Bridge, move
millimeters a
initiated.
year.
Do you
of
see that?
growth.
You see that watch lead? The green.
Was that?
See the green worm-looking creature in the middle of the frame? I just saw it moving around.
Yeah. Yeah, there is something there. Wow.
Yeah.
I don't think I've ever seen anything quite like that.
It's like a two-headed worm.
Yeah.
Interesting color too. We don't.
Let's
see
come out
that
partial
very
video.
often.
Come out partial, and then I'm going to touch down in a bit. When I do, hold that.
Gonna maneuver to get a closer look at it.
A bit when I touch down, he retracts.
Our pilot Dan had a very good eye to see that
Oh, he's staying out there. Okay, go ahead and zoom in.
green worm-like creature against all this other coral rubble.
Hold that.
Oh,
It is
There
a
he goes,
worm.
there he goes.
Look at that.
Wow.
Not quite all the way. Okay, let's come out.
Huh.
That was great, Dan. Thanks.
That's a new one for me.
And speaking of our pilot, I was going to ask them to introduce themselves, but they're actually having some discussion up there, so we'll wait. There's a shift change as soon as we start.
So sorry we didn't get to introduce our first round of pilots for the day. I wonder if that worm that we just saw would be a spoon worm, an Echeurin. So they're annelid worms like earthworms, but they've lost their segmentation.
Okay, pilot, 80 degrees on the rotator, and I'm still moving.
Copy. Hey, watch lead, we just had a pilot shift, and we're starting to move again.
Roger that. Thanks a lot. Our new pilots are settling in, and we're on the move again.
Once we get the new co-pilot in, I can describe what we're doing.
Oh, I'm going to stay here for the next shift.
Yeah.
Oh, you are?
Yep.
Okay.
Let's do a snap right here.
Oh, there's the fish that swam out of view earlier. Oh, that's a nice...
Isn't that beautiful?
Yeah. Love the blues and greens. The black saddles on his back.
Right, come in tight.
Oh, that's gorgeous.
Wow.
Okay, let's come on out. I'm going to move along.
I don't think we have any fish folks on our chat room to help us with the identification of that. Perhaps Ken Sulak can review the footage if he pops online today.
Let's get this
Give
fish
us
as
some
I come by.
information about the species.
And that's the Lamanema melanurum, the reef codling or coral hake. That guy doesn't have as much black on his fins as the ones that we were seeing before.
Right, let's come on out. Right, let's get this guy.
And a rat tail, Nizumia species. I think there's a couple in the area, so I won't even venture a guess on the species name. And Alexis Wenning from Temple University suggested that the green worm we saw might be an acorn worm. Acorn worms are Enteruptinusta, and they're hemichordate inverts.
Come on out.
90 degrees.
Pushing ahead.
I noticed that the sonars are both set to 50 meters on D2 and Sirius. Would you like those to be adjusted, or are you happy?
You feel free to adjust them.
Copy that.
There's not
Mine
a
is
whole lot
good.
to look at. Pretty flat.
Easy enough. I'll leave them how they are.
No, there goes another squid. And
ROV nav bridge, move complete.
Good copy, Bridge. Thank you.
And it looks like we have our first urchin of this variety. We saw the pencil urchins earlier.
Go ahead in.
And I would imagine this is either Echinus or Gracilochinus, are the two urchin species that are common in this kind of habitat.
Nav, after he's done with this zoom, we can call in another move.
Yeah, I was just going to mention that. You want? I see that you guys have been doing 250.
Yeah.
Do you want me to shift it to 240 now?
Sure.
Whatever you need to do to get to your waypoint.
Copy that.
There's not a lot of...
Seems pretty flat, so
Yeah.
I'll just keep calling in moves.
For us, it doesn't really matter.
All right.
Just get us...
You see a lot of the
Go in the direction you need us to go. It's coming tight.
remains of corals, solitary corals, the cup corals.
That's max.
But most of the branches, of course, are the Lophelia corals, the branching framework coral that
I'm going to call in a 30-meter
we
move,
expect
pilots.
to see a lot of
good.
as we climb
Come on
up
out.
this
At,
ridge and up onto the
240.
top of the mound.
So it's only 10 degrees.
And when you
Actually, you're
start
lined up
seeing
with it now.
rubble like this down low,
So co-pilot,
it's
if you want to
fallen
swing
from
around.
someplace, and it
Okay.
usually
Bridge, this is
means there's
ROV
gonna be
now.
a lot
Go ahead,
on the
RV now.
top.
I'd like to call in a move of 30 meters at 240 degrees, a speed of 0.2 knots, please.
That's 30.
And in case weren't able to join us
meters,
earlier
going
this
240,
past
speed
week,
decimal 2.
we
Good
dove
copy,
on
Bridge. Thank you.
3 mounds of similar structure that were formed by the scleractinian corals over thousands of years, and the corals will settle on something initially, some kind of little bit of hard substrate, and then
ROV
colonies
nav,
grow
bridge,
and
move
grow.
initiated.
And they can
Good
grow
copy,
on
Bridge.
top of
Thank
each
you.
other. They can also, one colony can fragment and continue
Yeah,
growing.
let's do
And
a zoom here.
As they do that, they baffle the sediment out of the water column. Oh, and look at that.
Looking right at us.
That's beautiful. Maybe Mike Vecchione can tell us which octopus we're looking at here if he comes back on.
Or if Stephanie Bush is in the area on board. She's also knowledgeable of these different cephalopods.
And Dougal Lindsay said that the colorful fish that we saw might be chlorophthalmus. Oh. Glad we're getting a good look at him before he runs away.
He has stalks above his eyes. I've never noticed that before.
Yeah, hold on the zoom for a second there.
Last week was cephalopod week.
Now go ahead
And this one?
you can go in tight now.
It's a beautiful shot. Especially with the curling of his tentacles.
Yeah.
Just using that as a platform to stand up.
Hey, Mike Vecchione here again.
Hi, Mike. We were hoping you might chime in.
Yeah, because you said octopus, I looked, and
Good. It worked.
It looked like it was another Granuledone vericosa,
Yeah, let's
which
come
we
on
saw yesterday, day before yesterday.
Right.
There was one in the mud before. There's another species down there that could be confused with it, and the way to tell is to get a close-up of the suckers. But the other species is Bachypolypus verdii. But this one, because of the bumpiness on the skin, I'm pretty sure it was a granoletony.
And are they fairly common?
Yeah, well, they're not uncommon. I mean, you don't see big numbers of them, but you do see them fairly regularly from about
Rotator
where
at
you
85.
are on north up to all the way up to Canada.
That's great. Oh, we
There's
have another.
I can't tell you a whole lot more about them because they're deep water species, and we don't know a whole lot more about them other than that, how to identify them.
Get this
and
fish.
that sort of thing.
Yeah, and that's true for so many of these taxa down here. We can maybe get an ID to a genus or sometimes a species, and then we don't know much about their behaviors or life histories, which was one of the things that makes all this observation so valuable.
We've got a student, we being Scott France and I, are working with a student who's going to be beginning in the fall at University of Louisiana. She's going to be looking at these bathyal octopods, the ones that are found at these slope depths, so from about 200 meters on down to a few thousand meters, because the octopod, benthic octopod fauna is very poorly known. So she's going to be
90
looking
degrees on
at
the
all
rotator.
of the observations that Okeanos has
do a
been
quick
doing
snap in.
over the years in this region at those depths and compiling all the information to look at things like habitat selection and reproduction and those sorts of things. So
Oh, that's wonderful.
It's coming out.
observation will contribute to that. So every time you see one, it'll be a contribution to trying to figure out the biology of these things.
That's great. And we were just zoomed in on a chain dogfish. It's the second one we've seen. It could be the same one, of course. He might be following us. Curious as to our lights.
Let's do a snap here.
Another black belly rosefish. Yeah, and Mike, that's a very good point. That all of this video, I'm sure, will be looked at by many, many scientists when they're looking for one particular organism. So we have the grand overview.
It's another octopus.
Oh, there's another octopus.
Come in tight.
Oh
It's
my
tiny.
gosh. It's a little baby.
I only have a moment
Unfortunately,
here.
I can't look and talk at the same time, so I'm going to go back to the video.
Okay, great. Well, thanks so much.
Well, he has
I'll
a
call
lot
back
more,
in if it's not a granoletony.
lot more ornamentation to. Oh dear.
A fish just knocked him out of the way.
Hey.
I want the spotlight.
I know.
Go away.
It's coming out.
There's enough room out there for everyone.
But when the paparazzi come to town.
This
Ooh, there's
move
a
should be
big
wrapping
one.
up here
Oh,
shortly.
yeah.
I'll let
Is
you
that
push
our
ahead,
first
pilot, and
redfish?
then I'll
I
call it
was
another.
going to say, it looks
come on.
like
Copy
a
that.
wreckfish.
Come on in, video.
Yeah, that's a big one. There may be another in
Now
the
hold that.
distance. There is.
Ooh, wow.
Look at him.
Kill
Redfish
lasers.
or snowy grouper? Or is that the same thing?
It is not the same
Not the
thing.
same thing.
And
And they're both of them could be here, correct? That looks more grouper-like to me.
yeah, it depends on how big he is. If he's
Yeah.
really, really big, it's probably a wreckfish.
Yeah, it's
But
coming out.
yeah, he's motoring around a little more than the wreckfish I've seen.
Let's get the crab.
Oh, we certainly are seeing a lot of fish diversity. And our first Emunida picta. I've been wondering why we haven't seen this guy. He's very commonly seen on a Lophelia habitat.
I'm losing you to the port
Yeah,
side
I
of the
see it.
camera, coming up a little bit. I mean to the starboard side.
Yeah, yeah. Hey, it's coming out. We get lasers back.
Do we have somebody who just joined the line?
I think they were leaving. The wreckfish has very spiny dorsal fins, and I think we saw that. I'm definitely not a fish expert, but and maybe I don't know. We'll have to. There were two, so maybe we'll see more. These are the. If it is a wreckfish, it's great to see them.
Either one. They're both commercially
Quick
important
snap.
fish.
Yes, of course, yeah.
Quick snap here video. Let's put one there. Let's get this other guy. Go ahead in.
Are there black belly rosefish?
Yeah, it's coming out.
They are a type of rockfish in the Sebastidae family. They are sit and wait predators.
I'm going to go ahead and call another move. Same thing, 30 meters, 240 degrees.
Copy.
Bridge, this is RV Nav.
Go ahead, RV-Nav.
Would like to call on another move. The exact same thing, 30 meters at 240 degrees,
ledge
speed
here,
0.2 knots, please.
30 meters, bearing 240, speed decimal 2.
Good copy, Bridge. Thank you.
We've been slowly traversing this sort of ridge-like feature of the mound, which is located on the landward side of it, and likely formed by the Gulf Stream
RV-Nav,
hitting
bridge
the
move
main
initiated.
portion of the mound.
Good copy,
and
Bridge. Thank you.
moving its waters around it and providing some good habitat and the distribution of the coral around the edges of the mound as well.
There's another Eumutida picta. We've actually seen these squat lobsters grab fish out of the water column and start eating. So they're definitely predators, and they often will be seen standing on top of coral, so they get a little more elevation so they can get up in the water column and grab small fish and other creatures that are swimming by.
Do a quick snap on this fish.
And another Nizumia rat tail.
Watch lead, this is Nav.
Okay, coming up.
Go ahead, Nav.
Just wanted to let you know that we have moved 100 meters since landing on the
There are
bottom.
a couple of
Roger
cup
that, thank
corals.
you.
You're starting, you start to see a lot of them now, the little yellow solitary corals. They're all at the base of this structure, so maybe that's a good sign.
That could be the genus Bathopsamia, the
I think there's
little
something to
orange
your starboard
cup
side.
corals.
A big fish.
You can see several Eumutida picta here. Getting it, wow, a whole bunch of them. Getting
Yeah,
into
I
a
might
little
have
higher relief
Gotten out of the way.
and more live Lophelia coral.
Yeah, they are. Yeah. All right, come on in, Ron. Bob.
Oh, here we go.
And
Good
some
look
more.
at
Look
Right,
at
hold
all
that.
of them.
Hold that.
Yeah, if they put their fins up, we might be
Yeah,
able to tell. Their
the
dorsals.
the snowy grouper
Yeah,
has
they're
a little
looking-
patch on the back.
Yeah, they're
too.
looking more like grouper. And wreckfish are quite solitary also from what we saw at the Charleston Bump, which is their
Coming back
spawning
your way.
habitat.
Let's come on out.
Snowy grouper, we're thinking that those are snowies.
Yeah, at first we were hoping that it was a wreckfish, but on closer inspection, does not appear to be.
Sure.
Little topography here. Some different mounds having grown through time. They haven't coalesced, or maybe the currents have prevented them from joining into one larger mound. See some of the sediments in the area too. Those are carried in by these currents.
The snowy grouper are actually considered vulnerable on the
Bridge,
conservation
this is ROV
status.
Nav.
Go ahead, RV Nav.
I was just curious if a bailout direction was discussed with you guys yet.
Yes, northeast.
Excellent, thank you very much.
Yeah, no problem.
We've climbed about a little more than 15 meters.
Let's do a snap zoom here.
RV Nav Bridge, move complete.
Good copy, Bridge, thank you.
Just come on in, video.
Nice shot of an anemone. Or is that? Yeah.
Again, pilots, once D2 gets pushed out a little bit further ahead, I'll
Many
call
of
them the same
those
move,
brittle
another 30
stars that
meters,
we'll
and then we'll
see
change direction
all
after
day
that, most
long,
likely.
every dive.
Copy that.
Tube worm. Several of them. You can see the brown coloration of the dead coral. That is from iron and manganese oxides precipitating out of the seawater. It coats them.
Some beautiful anemones in here. And more brittle stars.
Okay, let's come on out.
There's a paper that some colleagues
Oh, there's a sponge.
published several years ago about brittle star damage. Basically, they looked at, took big
Let's get
areas
the
of
sponge.
this kind of habitat and pulled out all the brittle stars, and about 60% of them had damage to their arms, and so they're probably quite a food source for many organisms, and they can regenerate arms, but those are very vulnerable, it looks like, when they're hanging on like that, and some fish or something else could come and munch on one.
Is this a glass sponge we have? One of the many we've seen in our adventure. At several different depths. We seem to encounter them everywhere in some form, different species, of course. Our
Okay, coming
greatest
up.
depth dive was at 3,300 meters. And we even saw them at that depth. Starting to see a few more of those sponges.
Navigator, I think we're ready.
Good copy. I will call on the exact same move, 30 meters at 240.
Copy.
Bridge, this is ROV Nav.
You mooted a picta?
Go ahead, RV Nav.
We'd like to call on another move, the
Okay.
exact same thing, 30 meters at 240 degrees, speed
We were
0.2 knots, please.
Yesterday
30
evening.
meters, bearing 240, speed
about
decimal 2.
Good
coral
copy, Bridge, thank you.
diversity and how it changes from dive site to dive site. And in this high Lophelia habitat,
ROV
Lophelia
Nav,
is
Bridge, move
basically
initiated.
the dominant coral.
Good copy, Bridge,
So
thanks.
we'll see high cover of coral, but it's mostly the one species. And that's quite different from some of the dives that we've done recently on scarps.
I think you're reaching the
where
end of
we
your
seem to
tether,
have
pilot.
a
Yeah,
much higher
I know.
diversity of coral types, coral species.
Ship is underway.
Thank you.
Copy.
Let's get this squat lobster. I think he's missing a leg.
Yeah, I think he's missing a leg. Again, these squat lobsters are a relative of hermit crabs. They're decapod crustaceans, so they should have five pairs of legs, and their last pair is modified and is kind of tucked up underneath the abdomen. But this guy only has 3 legs on the side that we're seeing.
2, I think, and one really tiny one.
And we have a pencil urchin.
It's coming tight. Or is that Max?
You can see where that leg is missing from. Might be starting to regrow a leg there. Actually, he doesn't even have his, I guess it's the pincher claws that are missing.
Yeah, he's missing.
This guy's having a rough time. He'll have to modify his diet for quite a while without those claws to catch the prey, which is what they usually feed, how they usually feed.
Alright, let's come on out.
When they have those claws, they're very protective or almost aggressive when we zoom in on them. They put those claws out, ready to fight. Here's another of the groupers.
wow. This one's
Let's get lasers
being
real
quite
quick.
cooperative.
There you go.
Near the lasers, use a big one. Those lasers are 10 centimeters. So he's
Let's
close
come
to a
in
meter
and see.
long.
That's a great shot.
Let's go in on the head.
Wow.
This camera is incredible.
Mm-hmm.
It really is, to be able to see the scales on a fish.
Yeah. Know if he had a little injury right below, right
Looks like
behind
he
us.
has a parasite on his fin a little bit, on his dorsal. Oh no, he certainly looks like a wreckfish. But I don't know the snowy grouper. I don't know either of them very well, but his coloration. Hopefully we'll get someone online who can review this and verify if it's a snowy grouper. It's more likely to be a snowy grouper, but if it is a wreckfish, we'd certainly want to know that.
Alright, that's coming out.
Let's see if there are any fishermen on board.
Looks like Ken Sulak has been feeding us some information about the other
Oh.
fish that we've seen, but hopefully he'll see
Yeah,
this
good.
soon too.
Good.
He's talking about the chain dogfish, Sclerorhinus redifer, and saying that they're one of the only sharks that can actually breathe while sitting on the bottom, while the rest of the sharks have ram ventilating during continuous swimming. In the dim light that we have at this kind of depth, the chain pattern on the dogfish is a really good disruptive camo.
Navigator, are we still moving?
I believe we have just come to a stop or are about to come to a stop.
Okay. We can call a move as soon as we are stopped.
Copy that. I'm going to change the bearing a little bit. It's going to be 225. So you'll be coming
Copy that.
15 degrees to your port.
Copy
See
that.
the ridge form that we are following. We're moving up this. We started at a depth of 450
ROV
meters
nav,
and
bridge
hope to
move complete.
ascend to
Gonna
Good
change
copy,
my
close
head
bridge.
to
to
375
pilot.
meters.
Pilots, I'm calling that in.
Jeez.
It is
Close to
225
the peak of this
30
large mound.
meters.
It's known as
Copy.
Bridge, this
Lophelia
is
Mound.
ROV nav.
We are off
Go
the
ahead, RV
coast
nav.
of
I'd like
Cape
to call
Fear,
in another
North
move.
Carolina.
It's going to be different this time. 30 meters at 225 degrees. Speed 0.2 knots, please.
At 30 meters, going 225, speed decimal 2.
Good copy. Thank you.
ROV nav, bridge move initiated.
Good copy, bridge. Thank you.
we're seeing a little bit more live Lophelia, which is the white coral that you're seeing. And a Venus flytrap anemone on the left. Some more hexactinellid glass sponges and Lophelia and Eumutida picta. And in Echinus urchin.
So the rockfish do snap.
Well, this is a very different color pattern than we saw on the black belly rosefish. I know that there's another score-painted fish that could be here. I don't remember the name of it, but hopefully Ken Sulak or another fish expert will let us know.
Hey, come on up. Let's get this right here.
Wonder if that's a very large hydroid and an Echinus urchin. But yeah, that looks like a hydroid to me. That's quite a big colony.
Come in. Let's come on in.
Oh, and a little baby shrimp